I'm a third-year PhD student at Hopkins Marine Station.
And I'm Grayson, I'm a junior at Salinas High School.
One of my big research questions is on the effects of nuclear radiation on wildlife, especially in Pacific Islands. In the 1940s and 1950s, the U.S. Navy did a lot of nuclear bomb tests in the Marshall Islands, and now I'm interested in going and looking at the animals that are still living there and seeing whether that radiation has effects on them and on their mutations in their genomes.
So I'm looking at coconut crabs, which are a native species to the Pacific Islands. They're the largest terrestrial hermit crabs you can see on the screen. So I'm comparing the size of the carapace has two kind of halves to it, and I'm trying to compare if they're symmetrical or not, and it could be if they're not symmetrical, which most of the times they aren't, that could be a result from nuclear radiation or some other mutation in the genetic code. The big takeaway from this internship would probably be just the experience of going down to the lab and being here and listening to Laura and hear what she's doing and how DNA research works, and there's different experiments and how an experiment actually works, how you conduct research properly, and it's really interesting because I got to see how actual scientific career works, I guess. Yeah.
So I grew up in San Diego, and I did my first internship when I was a junior in high school at UC San Diego, and so it was an internship in a neuroscience lab, and it was the first time I ever got exposed to any sort of science research and what that meant and what it meant to work in a lab. And so that first experience for me was a really great way to see, oh, this is something that I'm interested in and something that I'd like to pursue after high school and see what comes of it. Working with Salinas High School students has been great. Grace is my second student working on coconut crabs, and she's been incredibly motivated and has found some really cool and interesting patterns and has made real scientific discoveries. And so it's not only a huge help to me, it's also really exciting and inspiring to see people making new discoveries after learning these tools just a few months ago.
My name is Hana. I'm a senior at Salinas High. I'm in Miss F's marine biology class. I started this internship here at Hopkins. When was it? December I applied. Something like this. I have been working here weekly with the help of my mentor named Sherelle. She's from Canada, really cool. She's not here right now, she's in South Africa. What I do is I watch these whale videos. Okay, this is called a tag. They put it on the back of a whale's back, right behind its blowholes, and it records whales swimming around. It stays on for a few weeks and falls off. Eventually they collect it and collect the recordings and data off of it, and I watch the recordings and data. We're trying to find out how much energy a whale needs in order to dive down, or how many breaths it takes. You would think it needs more breaths to dive longer, but that's not actually confirmed anywhere. Like, nobody actually knows this. So we're trying to, usually when you use more energy, you breathe more. So we're recording how much breaths it takes, how long of an inhale, how long of an exhale it takes, and hopefully that will translate into how much energy it uses and how much energy it needs to dive. Or, but also, this is also a mystery, when it dives, does it take a lot of breaths to save up for the dive, or does it dive and then need a lot of breaths afterwards because it just wasted a lot of energy? It takes a lot of energy to go down and feed for long periods of time. Whales can hold their breath for up to 30 minutes, but usually maybe like 5 minutes is what I've been seeing in the past few videos. Right now, the video I'm watching is a South African humpback whale. They're all over. My mentor is in South Africa putting these hopefully 3 of them on 3 different whales, and we'll get videos back, which will be edited so that I can watch more videos.
Hi, I'm Julia, and I'm a 4th-year PhD student at Hopkins.
I'm Madison Lockenwally, and I'm a junior at Salinas High.
So in my research, I try to bring together a bunch of different types of data to understand trends and changes in fisheries. And so in my research, that involves looking at long-term catch data, incorporating environmental satellite data to understand what's going on in the ocean, and also talking to the fishermen themselves and doing interviews. And one part of this project that I'm working on with some of my lab mates is digging through archival California newspapers to look at historical seafood ads to understand how what fishermen are catching is influencing how seafood is being sold in California and vice versa. And so that's the project that Madison's been helping us out with. I've done a couple internships. I worked in a nanotechnology lab in high school, and I was also an intern with the National Park Service doing scuba diving research. And I think an internship is an awesome way to try something out. A lot of times with different types of research, and especially with science, there's really no way to know if you like it or if you're good at it until you get a chance to try it. And internships like this are a really wonderful way to get that hands-on experience.
This internship has really led me to be more interested in the marine bio programs that the future college I might attend would have to offer. This experience has helped me a lot with data entry, my research skills, such as using different databases, and I've also
been... Yeah, I'm Ben Burford. I'm a 3rd-year PhD student at Stanford University, and I'm from the area. I grew up out in Kashawa, if anyone knows where that is, out in Outer Carmel Valley. So, born and raised in the Monterey County. Let's see. Well, I went to Cal Poly to do my undergrad, so I never really strayed very far from home. And I studied field and wildlife biology there. It's actually mostly terrestrial stuff, believe it or not. So like birds and plants and bugs and mammals and reptiles, all sorts of fun stuff. And then after undergrad, I was really lucky to get an internship at the Monterey Bay Aquarium Research Institute, where I got to actually do novel research in deep sea biology. It was really fun. It was such a great internship. And that internship really sold me on the research side of things, because actually during undergrad I did not do a whole lot of research help. I think Ian is way ahead of the curve on this one. I was not really sure what I wanted to do in undergrad. I didn't seek out a lot of research internships as an undergrad, and I do regret that definitely. But what I did do, which really pushed me on the career path I'm on right now, was I actually TA'd as an undergrad at Cal Poly for 3 years, and actually getting the chance to teach undergrads in college is what really made me want to become a professor in the first place. I really liked that age group. I mean, people they're paying to be there, so they tend to be pretty engaged, and so it was a lot of fun. It was a lot of fun teaching undergrad biology and chemistry and stuff. So then afterward, that's why I sought research internships like Mbari's, because I thought, well, I should probably get some research experience before applying to grad school. But really Mbari, the internship at Mbari, I can't thank them enough, because that's what got my foot in the door in the marine biology field and made me a better candidate for graduate school. And as you go along through internships, you meet people, you have experiences, and it's ultimately the people you meet that will get you where you want to go. So it wasn't necessarily the skills I learned at Mbari, although those were very helpful. It was my mentor at Mbari that helped guide me to grad school, and his letters of rec and their letters of recommendation were what ultimately pushed me through the door at Stanford. Like, I never thought in a million years that I'd get to be a graduate student at Stanford, but I was very lucky. So it's a lot of luck involved. So I'm very happy to have the opportunity to have interns like Ian, because it is going to be such a huge part of my job as an academic, because that's really what I want to be, professor. So I'm really excited to have the opportunity to work with Ian and Ashlyn as well. It's been so fun, and it is the most rewarding part of my job. Like, that and teaching are the most rewarding part. Like, publishing papers is nice, but helping students is even better. So.
All right, I am Ian, and I'm a junior at Salinas High. All right, so here at Hopkins, he's given me some videos of squid that he's caught in trawls, and I analyze them, and I piece them together, and I look at how they're all angled towards each other, and essentially we're trying to support the idea that squid may school. A lot of big takeaways, like, this is great. I've mainly become really inspired to continue doing stuff like this after the internship. I mean, this is amazing. This is exactly what I dreamed of when it comes to scientist work. It's amazing. It's not too crazy. All the people are great with one another, and it's a great experience. I am definitely from marine biology, and if not marine biology, then just regular biology, something specific in there. But definitely, I want to continue doing work similar to this.
So we have a wet lab facility here where we basically get seawater flowing through these pipes up here, and then we can distribute that to a bunch of different tanks in here. And so I do a lot of experiments in the wet lab. But if you come over here, you can see we still have some squid. We caught these a couple weeks ago. Pretty much, I go out almost every week and go catch market squid. So these are all that are left of our group we caught last week. You can see them. These are California market squid. So you probably, if we walk out to the point, you'll still see some boats.
Yeah, to say I saw some squid boats out there.
Squid boats. Well,
yesterday.
This is what they're after. If you go to any restaurant locally and get calamari, like little rings of fried squid, this is probably the species you're eating. That's ridiculous. These little red crabs on the bottom are pleuroncodes, so they're called pelagic red crabs, and these guys don't come up here unless the water's pretty warm, so like during a pretty strong El Nino. So these guys, I think, washed up on the beach in late 2015, early 2016. But anyway, back to the squid. They're a really cool species. They don't live very long, so these guys are at the tail end of their life, and they're probably around 5 to 6 months old. So that's part of the reason why I'm so fascinated by squid. They do everything that your classic fish does in about 1/10 of the time. So you're ecologically similar fish. So when I say ecologically similar, I mean in terms of what the fish eats and who eats the fish. You could say that sardines and anchovies are really similar to the market squid. The sardines and anchovies, they're favorite. So the anchovies are the smaller, darker ones, and the sardines are the larger, more silvery ones. They eat, their favorite thing is small crustacean prey. That's what they like to eat, and then they're eaten by a whole host of things that we think are cool, like whales and dolphins and big fish and all that good stuff. But fundamentally, there's a huge difference between the squid and the fish, right? I mean, apart from just their looks, the squid is going to do everything that these fish do, but it does it in a fraction of the time. So these sardines right here, they probably reach maturity maybe at around 1 year or 2 years old, and they can live, the maximum age ever recorded for a sardine is 25, but typically they're living 15 years old. The anchovies are a little bit younger lived, so their max age is they'll push 10, but usually they're around 5 plus. But contrast that with the market squid that we looked over in that tank that reaches this very similar size, does a very similar thing. It takes about 5 to 6 months on average for them to reproduce. So very similar goal, totally different strategy, and so that's why I think squid are cool. That's what a large part of my research focuses on is comparing and contrasting squid and fish, and you can learn a lot about evolution fundamentally by doing comparative work. So anyway, we got fish, we got squid. These squid, I mentioned that they reproduced, and so they're what's known as semiluparous, which means that they reproduce then die. They have one big reproductive event in their lifetime, and nest of female squid. Over here in a tank, we have a bunch of eggs that were laid by the squid from this most recent group that we caught. You can see the ones that are further along in development are hung. You can see the eggs are a lot bigger inside, but each capsule holds, a couple hundred eggs. And these capsules down here are a bit, they're not as far along in development, so you can't see the eggs as clearly. But at some point soon, it might even be now if I had some light to shine in there, but you'll start to see the little squid develop inside the eggs, and they actually, their eye spots come in pretty early on, and so it's very cute. You can see these little squidlets in there, and you can see each egg has a little pair of eyes in it, and then eventually they hatch, and they're like about the size of Rice Krispies, and they kind of squink around. They're very adorable. And yeah, so those same eggs are probably on the floor of the ocean, not too far off the marine station here, where these squid like to spawn. Were the ones that we caught here. And yeah, so we're constantly bringing squid in here, doing experiments, bringing fish in, doing experiments, and this is where a lot of the work happens, is in the wetland. Yeah.